Hello, everyone. Good morning. So my name is Ivo. Just that you know, I'm a bioinformatician by training. So maybe here and there, some of the examples that I'm going to give might have something to do with bioinformatics or with marine things, marine microbiology, but most probably you won't even notice it. So for me, today is a big pleasure to present general concepts of data management and data literacy. And as I already told Daniel about a minute or two ago, when I was preparing for this, I realized it's a really huge topic. There are too many facets, and I only have 1 hour. But I still find it very important to bring in several important messages. So I'm going to go in some places maybe a little bit faster. You're going to get the slides anyway. They're going to be on Zenodo. So if it's all in all too fast, please raise your hand. And yeah, that's basically it. So we're going to use the chat in the first couple of slides. So get prepared to throw out some answers. So there's a tiny bit of interaction going on. So we are talking about data, and I believe Daniel showed you some motivation why we need to care about our data. So different aspects, and I want to bring one more additional aspect to why we care about data management, which right now is still a struggle for a lot of us. So basically, the idea is, you might have seen this before, is that data is the basis of anything, almost anything we do nowadays, whether it's industry, whether it's science. So we take the raw data, we produce some kind of information out of this, we try to generate knowledge, and then make some abstractions to get wisdom out of this. So I don't know how the last part exactly works yet, but when I figure it out, I'll be sure to let you know. Now, how is data produced? So how much effort and time and money goes into producing data? This is the aspect that I want to bring in. So I'm going to give you about 15 seconds to throw out some numbers in the chat. What do you think is the general research and development funding in Germany, not only in science, but also in industry? So just throw out some numbers. I'm going to give you 10 seconds. Take a sip of my coffee, and then we'll see if you get in the general ballpark. There's no right or wrong answer. I'm just trying to figure out who has read the same information and I did. Any numbers in the chat? Feel free to open the mic and shout them out if you want. That's 10 million. That's about 50 million. Not enough. Okay. Since we're a little bit pressed for time, and I'm not going to take this long on this one, close but not quite. So it's about 160 billion in 2021. Okay. So the numbers are from there's something called the Funding Atlas made by the DFG, the German Research Foundation. It's linked down there. It comes out every 3 years. And it's a very interesting read. So you can see where is the money coming from, where is the money going to. Keep in mind, this also includes industry. It's not only research, right? So 160 billion. And I've been doing this I've been reading the Atlas for the last almost 10 years, and I can tell you the number is only going up. So this basically means we're putting a lot of money, even if this is not the whole number, a big chunk of that is going directly or indirectly into producing good scientific data. Now, the next question is directly related to you and your work. So do you have a gut feeling estimate how much time in percentage of the overall time you work on a project is going into discovering and reusing multiple data sources? So basically figuring out, what data do I need to compare my data to? Where do I find it? Can I use it? Is it licensed the proper way? Are the parameters the same? Are they in the same units? So how much time would you say go into this? Throw out some numbers in percentages in the chat again. 10, 15 seconds for this. 10, 25, 30, 30. Okay, very good. 60. Excellent. So those that are below 50%, if that is really true, I applaud. There is still this number that was calculated in the US based on surveys, a little bit better done than this one that we just did right now on early career scientists. So basically PhD students, early postdocs. And they said the effort that goes into this is close to about 80% of my time. So and we want to improve this. So why am I showing you exactly these two numbers, money and time and effort spent? Because these are additional aspects of what we put into generating the data, generating the information and the knowledge, especially in the area of biodiversity, ecology, environmental science. And it's taxpayers' money and it's your time and effort. So we should care about this on top of all the other aspects that Daniel showed you this morning. So it's kind of a protection of investment, taxpayers' money, and your own effort. And with that, we can start at the basics of research data management. So first of all, what is research data? We all have an intuitive understanding of this. But to put in a little bit of a definition to it, is any information that has been collected, observed, generated, or created in the undertaking of research. And that has a really wide range, so I'm not going to read all the points, but all the measurements data, observation data, laboratory notebooks, photograph samples, data files, even the algorithms, the scripts, the programs that you write, that you want to analyze or visualize the data, that all belongs to it. Now, there are different types of research data. So there's, of course, the primary or the raw data. That is what you collected, what you observed, what you generated. But there's also something very important, that's the metadata. And we're going to circle back to this because the metadata gives your primary data context on how to interpret and how to use it. And there's, of course, secondary data that are derived from your primary data, either by filtering, selecting, correcting, aggregating, creating new data sets, quality control, whatnot. And there's, of course, general research information like administrative information, who is involved in the project, running times, financing. We're not going to be touching on that today. Now, research data management is the process that involves the active organization and maintenance of the data throughout the research process. I would say including its publication, long-term archiving, but in some cases, even beyond that. We'll see if we have enough time to actually explore a little bit more of that concept. What happens after I've published my data? Depends a little bit on the time. Now, we've all heard about a good scientific practice. I'm not going to play the game by show of hands who has read the booklet of good scientific practice of your institution. That actually, most of the time, that has a yield of about 2%. If you haven't read the good scientific practices of your institute, please do so. You're actually required to do this and to know them and to apply them in everyday work. However, if you really pay attention when you're reading, you're going to figure out that a lot of these are very generically formulated. So in a lot of cases, you don't get very much help out of this. What am I supposed to do in my everyday work? So people start doing the science, and then in about 2, 3, 5, 6 years, and they figure out, oops, there we got some problems. So I cannot get some data because the person who produced it is not accessible anymore, either because they're retired, are dead, or just left the field. They don't care about it anymore. The formats are not readable. The units are not clearly defined. The structure is a mess and nobody can explain it to me, and so on and so on and so forth. So the good scientific practice on its own is, from my perspective, definitely not enough. But there's, of course, something on top. The guidelines of the German Research Foundation is also dates back, like the Safeguarding Good Scientific Practice, the memorandum dates back to 1998. And there is the Guidelines of Handling of Research Data as a recommendation already from 2015, which is also not that long ago. And there is a specific part or a specific add-on to this, the Guidelines on Handling of Research Data in Biodiversity Research, which for this group of people here, I think is the most important one. If you haven't read this, please do so. It's not so lengthy. It's an understandable text. And last but not least, I have to move some windows around here just to make sure that I'm talking about the right thing, is that the guidelines for handling of research data are now binding since 31st of July, 2023. So it will be good for you if you know exactly what is in there. You will need to do it because it has a code of conduct, includes the statement on the handling of the research data, the long-term archival, open access, reproducibility documentation based on standards and quality assurance. But as I said, a lot of this might be still a little bit generic, and this is why we're going to go straight into the FAIR data principles. So the FAIR data principles have been around for about 10 years now as an acronym, even before some institutions were using them to guide their work. The stands for findable, accessible, interoperable, and reusable. And what I always recommend anybody and everybody who has to work with data in science is please read the original publication by Wilkinson et al. It's very short, it's very well written, and it really makes sense for you to understand what is everybody talking about instead of just always relying on interpretations of somebody else, including the one that I'm showing you today. And part of this publication is, for example, this small box. Don't worry, you don't have to read all of it right now or even understand all of it right now. Are the guiding principles themselves. For example, that in order for data to be findable, one of the guiding principles is that data are assigned a globally unique and persistent identifier, that they're also described with rich metadata, and so on and so forth. So this publication is definitely the source to go to when starting to learn about FAIR. Okay, so if you start Googling FAIR data, you're probably going to get some good resources. You're going to get a lot of interpretations and explanations what FAIR is. One of the important things from my perspective is to also include the information what FAIR is not. That helps you understand what FAIR actually is much, much better. The first and most important thing is FAIR is not a standard. There is no standard for FAIR yet. So these are guiding principles. And if somebody talks to you about a FAIR standard, please ask them if in the meantime somebody has developed a standard or this or not. Don't take it for granted. FAIR is not the same as open data. So data can be fair and still be under some kind of control. My favorite example is with observation data of rare species. So when you publish metadata and data on rare species, the thing that you should never ever do, or the publisher of the data should never ever do, is disclose their exact location, because most probably the next day somebody will try to go and find them and take them out of their environment. So this is only one good example of when the metadata can be published, but not all of it. There could be some kind of access control saying, we know that this species has been cited somewhere in the general area of Munich, for example, but the exact locations are only available upon request and a validated proper interest. So you have to sign some documents, you have to contact somebody, say you're not going to disclose this location if this is really pertinent to your research. So data can be fair without being completely open. Then FAIR is a spectrum. So it's not a quality, but a quantity. So you cannot say data is either fair or not. Karen Goebel, who's very active in the FAIR community since the very beginning, calls this 50 shades of fair. So we have to think as a spectrum, which is also important for your work. If you like the ideas that we present today and you say, I want to make my data fair from tomorrow on, don't try to comply with all the regulations or all the guiding principles from at once. That will be rather difficult, but go step by step. Take the metadata first, take the DOI, and so on and so on and so forth. Just get better step by step. So FAIR is not only for humans and not only for machines, but for both. That's very important. So whatever we can do with all the advantages of FAIR data, machines should be able to do that as well. We still have to teach machines how to do that for now. That's okay. But there are some technical aspects when implementing the FAIR data principles that we need to observe in order for this to work. Most probably not part of your daily life in this case. FAIR is not only for life sciences. So the original team that wrote the paper that made FAIR famous came mostly from the life sciences, but they made a very good job generalizing the idea so that it also can apply to almost any discipline, including nowadays also just software, any kind of software. Last but not least, just to make the list complete, so FAIR is not the same as RDF or linked data or semantic web, so some kind of technical concepts and frameworks that you can meet out there. So they can be used to make data fair or fairer, but it's not one and the same. So you cannot say, I have an RDF representation, so my data is fair. That doesn't work this way. Now, just a quick remark, because humans like to classify things and to quantify things. There are different approaches to trying to quantify the fairness of datasets and repositories. And it's called the FAIR matrix. So one of the working groups is from the RDA. And of course, the usual suspects around Mark Wilkinson have wrote also a nice, easy to read paper on how the FAIR matrix could work or should work. I recommend you have a look at this. And there are several tools out there that try to implement the FAIR matrix and that to make it accessible to you in the sense that, for example, this tool Fuji that was implemented by the Pangea team, which they work on the same floor as we do from the GFBio FO, you can go there and you can just put in the URL or the DOI in the best case of a data set, and the tool is going to try to assess the fairness of this particular data set. And of course, there are studies that try to make a meta-analysis of this and go, let's assess the fairness of all the data sets in a certain repository and give a metric for the whole repository, which I personally think is not the right approach to do, but this kind of work is definitely ongoing. So maybe if you want to try to see how fair is one of your older data sets compared to the next data set you publish, you can use it to kind of check your own progress, for example. So why should you care about FAIR? Well, if you're concerned with FAIR, if you're working along the FAIR guiding principles, I would generally say you're almost certainly complying to the good scientific practice in general. Still, read that document from your institution and from the DFG. But also we have a mounting evidence that the better you treat your data, the better you publish your data, better your article acceptance, the better the data reuse and citation. Proposal funding is a big thing. You're going to see also in the DMP lecture that most probably I will have to take over today is you have to show that you're going to take good care of your data. And there's also something about the compatibility with future infrastructures. So you have heard about the NFDI in Germany. There's also the overarching initiatives about the European Open Science Cloud. And all these infrastructure can only work if the data is well described and findable, accessible, and interoperable. The last 2 points. So if you get tired of doing analytical research, there are always job positions related to what we do. They have different names and different profiles, from managers, stewards, custodians, librarians, all and about data. So you can join the dark side. We have cookies and more. But the point that I also would like to make that I think should be pertinent to this group, and I'm hoping that you've already at least heard of this, is keeping your research legal. So basically avoiding biopiracy. And yes, biopiracy is a thing, and it basically means when somebody from a better developed country goes to a less developed country and explores the genetic resources or any kind of resources, makes use of this, gets some kind of benefit. If they don't pay any tribute to that country of origin, that is just biopiracy. You're stealing things, whether it's knowledge, whether it's data, whether it's genetic material or something like that. That shouldn't happen, right? And in terms of genetic resources, oh, I see that something went out of the way here. I'll fix that afterwards. It's called the Nagoya Protocol on Access and Benefits Sharing. I hope that you've at least heard of this. because it's already ratified and entered into force in 2014. It's ratified also for Germany and over 100 countries, as far as I'm aware at the moment. And the objective is to make sure that biopiracy doesn't happen when it comes to genetic resources. It basically means what you need to do when you want to go sampling in another country. You have to do two things. First of all, you have to familiarize yourself with the local laws on research and sampling, especially for people coming from abroad. That's point number one. And then you have to make sure, has this country signed and ratified the Nagoya Protocol? Because that puts another layer of meaningful documentation on top. So Nagoya, important point, Nagoya does not replace local law ever. Please keep that in mind, but puts some additional mechanisms in place if you manage to get something meaningful out of the genetic data from another country, whether it's a product that you can sell and make money or it's just basic knowledge. There are mechanisms to pay tribute to the originating country. Many different ways. It's a jungle. It's difficult to navigate. That's true. If you've never done it before, you're probably going to be a little bit overwhelmed. You have at least two possibilities. So with an institution like iDiv, I suspect, and I'm sorry that I don't know in this in advance and I didn't research it, there might be like a Nagoya officer or a person that actually deals with that topic and can help you navigate the legal jungle. So if anybody knows if the iDiv has this kind of position or person available, please post it in the chat so that the others know as well. And then I can also learn it and use it next time I'm talking to somebody. The other possibility is there is this Central Nagoya Protocol hub that is set up at the DSMZ together with the, and now I have a problem with translating, the Federal Agency of Nature Conservation, the BFN, Bundesamt für Naturschutz. And they have information. I'm not 100% sure if at the moment they do also consultancy, but if you don't have anyone at your home institution, you have a better chance of contacting the Nagoya Protocol Hub and get some help in some form. Now, let's get a little bit more real about the day-to-day handling of research data. So I'm going to talk a little bit or use the so-called data life cycle as an instrument to structure what I'm about to say about what happens on a day-to-day basis. So the data life cycle is something that exists in different forms and definitely colors. So these are just our colors from NFDI for Bio. And there might be shorter versions, there might be longer versions. I think Daniel knows his way around the data lifecycle much better than I do. But this is the one that we've used for the past 10 years, more or less to structure our work as first in GFBio as like a national network of service providers, and now also in NFDI for biodiversity. And we'll see if we need to adapt it. At the moment, this is how we view the world or how we view the life cycle of data. So it contains 10 steps. It always has different facets, or the depth facets are different depending on your exact field or subfield. But generally, we say you start with a plan. You start with a plan, for example, a data management plan. Then you collect and collect your data, generate your data, assure the quality of your data. Then you describe, so you add metadata, for example, link whatever you used as description to existing terminologies. You submit your data for long-term preservation and publication to an established data archive or data center. They do most of the preservation. You do the preservation and the sharing during the project lifetime with your colleagues. And then you can start discovering other appropriate data, integrating, doing the analysis, and then publishing. And now please note, when we say publishing, most people will think publishing of the journal article, but we also mean this is the latest point where you should also publish your data in its own right. So you're going to see this concept, I think, more than once during this week. is that we propagate the publishing of data as individual units, not as um, an appendix to a journal article, for many different reasons. And what is see already here is that how it pertains, and also some of the topics that you're going to see this week from us. So, for example, the data management planning, the licenses when it pertains to data publication, and so on and so forth. Now, let's talk very shortly about the planning, because you're going to get a little bit more information later today, maybe on Friday, is the data management plan. That should be a formal document that basically explains how you're going to handle the data during your project and also once the project is finished. It should save you time. It should make you plan in advance. So if you're missing resources, if you're missing expertise, you're aware of it ahead of time, and it increases transparency to your work. So basically, personally, I propagate always publish your data management plan on Zenodo the same way that you would publish your project proposal, of course, without finances and home addresses of your partners. And it's a funding agency requirement. Until now, as far as I'm aware, a little bit shaky on how required is it exactly. Maybe my colleagues can help afterwards if they know the exact state. I think it's not absolutely mandatory yet, or at least people don't feel it this way, but it's definitely going to be absolutely mandatory in the near future. We're working very hard to get there. And the main elements of what the DMP does is a checklist. So who's responsible, what kind of data is going to be there, what kind of metadata is going to be used, how is this going to be captured, what kind of policies and agreements for accessing and sharing are there, what is the midterm storage, what is the long-term storage be organized like, ethical and legal compliance, costs and resources. So basically anything that pertains to your data. And what we do is we already have, for example, one example DMP for biodiversity in the area of biodiversity that's open, it's published, you can have a look. And this is what generally is going to happen in the practical part of the DMP session, either today or tomorrow. You can try to generate your own DMP. But I'm not going to spend more time on this because we have a whole session on that. Now, when it comes to collecting data and documenting data, the first and most important thing is be systematic in anything you do. It's better to have a suboptimal system than to not have a system at all or change the system every second day. So think of a system beforehand, optimally apply it, stick to it. So name things the same way, standardize your sampling protocols. That's something that you do already anyway. Determine how your files are going to be named. So make out a schema for this, write it somewhere, share it with your colleagues, put the time in to make sure that people follow it. Document metadata as soon as possible, basically immediately. The one thing that you cannot recover is missing metadata. We see this in the archival and publication process when people come and say, I want to publish this data, get a DOI, and then we start asking for metadata and we figure out was either not captured or this was captured in a way that is very difficult to retrieve. So your metadata is just as valuable as the raw data itself. Now, a few very, very practical examples. These are examples of best practices. They're not the only valid way to do things. Okay, so please be aware of this. I don't know how many of you use a Unix operating system. So for me, some of these, for example, apply to 100% naturally since 20 years. A few don't apply at all because I have a different system, which is just as valid as this one. So take this as a pointer. If you don't have anything, this is a good place to start. But then you can develop your own, you can talk to your IT, or if you have a data manager or a bioinformatician or somebody with a little more technical acumen on this, you can develop something that better fits the needs of your project or your working group. So keep file names short and descriptive, of course. If you use abbreviation and acronyms, do so consistently. Again, document this somewhere, make sure everybody who's involved knows what they mean. Use ASCII characters. If you don't know what it is, then probably there's a very little chance that you're gonna make a wrong decision here, but try to avoid spaces and special characters that are listed here. I can tell you a reason why. If you're working on a Windows machine, which I would guess at least half of you are doing, you might be very well used to just typing spaces in between file names. As soon as you send this or use this on a Unix operating system, it's probably going to be somehow okay, but the file names will be different. And if you take the next step and you develop scripts that are supposed to run and you develop them for Windows and somebody tries to run them on a Unix system, that's not going to work out at all. Okay, so that will be just from the practicality a bit why. If you use sequential numbering, it's generally recommended to use some leading Xerox, so the so-called padding, so that it has the same one has the same length as a number as 100. And if you need to rename files, do this in bulk. So for Windows, I guess there's this bulk rename utility, but for Unix operating systems, there are very easy command line tools that can actually do that for you. But the thing is, don't rename things by hand one by one. That's just error prone. One of my favorite topics is version control. So keeping the original version of the data file same and save. Also about the metadata and also about documentation. So I would like to extend the concept here a little bit. So we can talk about the data itself, we can talk about the metadata, and we can talk about any kind of documentation to it. So like your protocols, what you did step by step, and so on and so forth. So first of all, establish a consistent convention. Again, document everything. There's no such thing as too much documentation, even if purebred developers will try to convince you otherwise. There's no such thing as too much documentation. And I highlighted one thing. If you have the possibility, use a version control software, something like Git. I'm pretty sure all of you have heard about Git. I'm sure all of you have heard about GitHub and or GitLab. I'm pretty sure you have GitLab at your fingertips at your institution. I would bet almost anything. If you ask me what you can learn, one thing that you can learn that's going to make your life better, my first recommendation would be Git in its very basic form. Git is very powerful. You can get lost in the details, but the very basics can save you already a big amount of trouble and help you a lot with versioning, or basically keeping track of what's changed to the data due to my the processing that I do, what changed in the files, in the metadata, what changed in the scripts that I did, what changed in the documentation. So I also use it on a daily basis, even though I don't write code myself. Right, so use Git. Avoid imprecise labels. So this famous final, I think anybody and everybody has done it. I've done it as well myself. You know this, it's final, and then it's final 2, and then it's final 3, and this doesn't really help at all. One recommendation that might help you in this case is to say, use a date. And use an explicit date when the file was last modified. It's not as good as using a version control software, but it's better than saying final 2, final 3. What you're going to do is describe the data. All you need to do is describe the data. Why? Because describing the data in the same way, so basically I'm talking about using standard community accepted terminologies, is making sure we all speak the same language. And my colleagues have found this very, really nice representation of what people think they're talking about when they only see part of the whole, right? Is it a tree? Is it a spear? Is it a wall? And personally, I like the explanation in saying, if I describe where my samples come from as the bottom of a hill in Bremen means something completely different than a hill in Bavaria, right? The highest position in Bremen, the highest hill in Bremen is basically the garbage hill that was artificially made. And that's not a joke, that's just the truth. That's the highest point in Bremen. So use a terminology that basically describes what is a hill, what is an ocean, what is the deep ocean, what is a meadow, what is a forest exactly, what is a different types of forests, and so on and so forth. And we have a very clear small recommendation here because one of our partners and members actually runs a terminology service specifically targeting the biodiversity community. So you can look through the terminologies in there and select the most appropriate one. If you don't know which might be the most appropriate one or how to use them, you can always contact us on any topics. I think you've heard this before. You're going to hear it 1,000 times this week, but if you don't know where you're going with this, just drop us an email or use the contact format and we can give some advice. Now, describing what you're usually describing or using the terminologies for is maybe not exactly the data itself. It should be in parts, but also the metadata. So metadata, meta comes from Greek, and it means like overarching or a little bit more abstract. But metadata is basically data about data. Recursive definition, I know, but it gives context to your raw data. Where is it from? What does it mean? How is it described? How was it collected? And the data is at least as important as the raw data, because if you don't have any kind of metadata, if you just have numbers, nobody can understand and interpret that in the future. And it's very important that metadata can be machine readable, right? For example, who created the data? What is the content? When was collected? Where? How were the data created? How do I understand this data? A typical example, we're not going to show you any videos today, I think, but a typical example is you get a table and the table has a header column, and in the header column, there are abbreviations, and you never know what the abbreviation is. That's not a good example. A good example would be to summarize the basic information about the data in a structured way to explain this is what this header means, this is what the unit is, and so on and so forth. Saying temperature is not enough because my colleagues from Pangea, you're going to see also a talk by them, when they curate data, they're going to ask you, is this temperature air temperature, water temperature, body temperature, or something else? Makes a difference. And if it's temperature, if it's 62, is this in Celsius, Fahrenheit, and Kelvin? So the unit also makes a difference, and that's also part of the metadata. So metadata can also get very, very detailed and very complex, but that's a good thing in this case. So here you see an example about the Darwin core. So this is based on XML, an extensible markup language. So basically the blue parts are, if you want, the metadata type. So this is also using it's a standard and it also has a terminology inside there. So basically this piece of text is not only telling you Museum of Vertebrate Zoology, but it tells you this Museum of Vertebrate Zoology, they're actually the rights holder, right? And the rights or the license, if you will, is Creative Commons license. So the blue brackets are putting context to all the text. And this is specifically important for two reasons. First of all, if somebody looks at this and it's not coming directly from your field or from your subfield, they might not be 100% sure what this is about. And the second part, remember, fair machines should be able to do the same. This is how machines would understand the data. They can interpret this. If you just put the text in there, a machine would, at the moment, still not be able to correctly interpret this most of the time. There are also other standards that are relevant for the biological fields. So there's also the EML, ecological metadata language. There's also the ABCD, access to biological collection data. So anything that goes in the long-term data centers, data archives, all the natural history museums in Germany, or almost all, work with it. And there's also my favorite, the minimum information about any sequence. As I said, I'm coming from bioinformatics, so most of my work has been centered around the mixes. Preservation. So I'm only going to show something about the aspect of safeguarding the data on your side. Otherwise, the long-term archival and publication that should optimally be done by a specialized data center, data archive that has been doing this for the last 30 years. We have a few of those on board. You're going to see how we communicate with them by the end of this presentation. But our recommendation for things to do from day one is, for example, open file formats. What does that mean? You don't need a specific software to read these formats, right? Excel is a specific format. It's a proprietary format. That's the correct term. Excel, Word, anything from Adobe, those are proprietary formats. You can only open them and read them correctly using a specific software. You never know if this software is going to exist in 10 years, if you're going to be able to afford it, if your colleagues are able to afford it. So think about cooperations with less developed countries or just groups that don't have the same means as you. So concrete recommendations you can see on your screen. So for tabular data, any kind of separated values, whether it's a comma, whether it's a tab, whether it's another character that you use in there, that's a flat file. Anything can, any program can open this. Plain text, HTMLs, RTFs, TXTs. For structured data, you have the beautiful XMLs, RDF. I would say also JSON is absolutely valid. And for images, definitely TIFF or JPEG. Again, these are starting recommendations. Depending on your field, subfield, you might find other additional, more specific file formats that are well established in the community. I think one of the examples would be when you go into modeling, when you go into like 3D models. So it's an image file, but it contains different kind of information. There are definitely things that you can find there that fit the recommendation but are not listed here. And the question is always, how do you store? How do you do regular backup? Do you have any data policies, institutional guidelines? And my recommendation at this point is don't try to solve this completely on your own. Ask for help first, whether you have a data steward at your institution or you have to talk to your IT. Please talk to your IT first if you are unsure and if you don't have anybody else to talk to. A little bit about one minor detail in addition about storage versus preservation. And preservation is more than a backup. So in this case, we talk about storage as a midterm thing. So during the active project phase, it can be on your local computer or server. Please try to use institutional infrastructure and not just your laptop. Laptops get broken, stolen, lost, whatever. So anything that is institutionalized, anything that has some kind of redundancy, technical redundancy in this case, is to be preferred. And let's say having at least 2 or 3 different copies on different media, one at a different location is also preferred. And again, this might take a lot of effort or organization. Talk to your supporting staff before you try to solve it on your own. On the other hand, the long-term preservation also aims or looks at the integrity of the data. There are long-term repositories, most or many of which with curation routines, and they can guarantee storage of long-term preservation of the data according to the good scientific practice, which the DFG basically set at minimum of 10 years. So you're obliged, the data that you've produced and used for scientific process and publishing has to be available for at least 10 years. And accessibility means data can be retrieved, displayed, and used. So basically, you can get it back if you need it. It has to be authentic, so it hasn't changed in any way, either willingly or unwillingly. There are also minor mistakes that can happen even in transfer that you're not aware of. So nobody is doing anything explicitly, but these things can happen. It's still just technology, but this needs to be verified. And the data has to be reusable for long term, independently of software or hardware decay. And this is circling back to the idea of open file formats. So the open file formats have a much better longevity than any kind of proprietary formats. So getting to the point of discovering, integrating, and analyzing data, I think I'll skip here an extra slide because I'm just going to show you a few tools and ideas we do at the end of how we think these things can be done. But generally, you're free to discover, integrate, and analyze your data any way you see fit. However, please remember to document everything you do, including versions of software that you used, including routines. The best thing you can do is to always script your routines so that scripts can be versioned with Git, for example, and they can be also published, for example, to GitHub as part of your data and metadata package. But I want to spend a few minutes on the point of data publication, right? So I said in the beginning, we promote and we think data should be published in its own right, not only as an annex to a journal article. And you should be able to, or you should cite data on its own and not only the publication that first described it. So publishing data means publish data in a trusted repository. The data should receive persistent identifiers. For example, a DOI is the most well-known one. And that DOI, or that persistent identifier, as the name says, it's persistent. It should be there forever. DOIs do that. There are other possibilities. There's a handle system. We're not going to talk about that today, but it should be permanent. So just a URL to a website, to an FTP server or whatever is not persistent. Nobody can guarantee this. The DOI system, the handle systems, they guarantee. So there is a official registration authority, for example, DataCite, that's part of the DOI consortium. They guarantee, that's their job. That's why they take money for generating DOIs. They guarantee that this DOI is not going to disappear and it's always going to resolve to whatever you said it should point to. And PIDs link structured metadata to the data set. So you can also put some metadata inside the DOI even before it resolves to your data. And you see here down here the structure of ones of a DOI as an example. So the first part is the resolver. So basically, which address do I go to in order to find out what is behind this DOI? Somebody sent me a DOI, said, hey, look at this paper. So how do I get there? What is behind this link that I'm clicking on? The third part is the resolver, and there are several international resolvers out there. So it doesn't matter which one you use, you're always going to get the same result. That's the beauty of it. The most famous one, and the one that I also keep in my mind so I can just go and use it, is dxdoi.org. I believe you can also skip the dx nowadays. And then comes the DOI. And the first part, so between the two slashes, is the so-called prefix. That's the code of the publishing institution. So 10, 1, 2, 3, 4. I forgot if that's an actual one, if that's from Zenodo. But so mostly you can sometimes recognize, okay, this is coming from Pangea, this is coming from Zenodo, this is coming from somebody else. A lot of them are numeric, so it's kind of hard to remember. And then comes the suffix, which basically means this is the unique digital object. So it's not just paper, it's not just data or metadata, it can be anything that is a digital object that is unique and that has been saved and registered by this DOI issuing institution. Okay, that's behind it. Why publish data? There are many different things. First of all, it's a good scientific practice. It's what we've learned from day one in science. You are required by all the regulations and all the contracts that you signed, also getting third-party funding, and so on and so forth. So it's kind of intuitively clear how we should be doing it, but what are we getting out of it? So you get visibility and credit to the data collectors if you publish the data set completely on its own, including credibility and reproducibility. It's required by funders and publishers. With the publishers, I have to say it has different flavors in the different disciplines and sub-disciplines. For example, with genomics, genetics, it's very clear it's almost impossible to publish a journal article without publishing the data first. In some other disciplines, it's depends on the journal, depends on the editor, depends on the reviewers, but the trend is also going more towards very clear requirements to publish the data and also make it available before the journal article goes online. So the take-home message from all these many points on general data management and data literacy is remember the fair data principles, at least in general. Please read the original publication. The idea behind FAIR is to improve how we use and reuse our data. I think there was a famous quote that I forgot how exactly it's phrased and who to attribute it to. So if my colleagues remember this, you can post it in the chat. But basically, the best discovery based on your data is going to be done by somebody else. So I'm rephrasing this. I'm going to figure out who said that. And by doing so, you're increasing not only the reproducibility, but also the credibility of your work. So imagine the impact you have on somebody else when you say, my proposal is online on Zenodo, my data management plan is online in Zenodo, including 3 different versions that it did during the project lifetime. And all my data is either on Pangea, on NCBI, some of it is on Zenodo, some of it is in a bioimage archive and whatnot. And you can see all of it and you can reuse all of it because it has rich metadata attached to it. That's a different kind of impression and impact. So the recommendation is try to apply the fair data principles as best you can. If you don't know how to proceed or where the best way is, of course, you can contact us and get some help in different ways. So just a general recommendation, maybe a service, maybe some kind of support, maybe even install a local RDM system. And of course, as I already said before, start with small steps to integrate into your routines and start with the planning, think about it, learn about it. It will save some time, spend the time up front. It will save not only time, but also it will save some pressure that might come on you later on in the process. Okay, I hope I wasn't too fast. I know I sped up somewhere in the middle. I hope that was still understandable. I'm going to show you just a few, very few highlights of the service catalog we have inside NFDI for biodiversity to help with research data management in biodiversity, ecology, and environmental science. And what I want you to do, what I want to ask you to do is, because this is 4 minutes, I could do probably a full hour only on the services themselves and still not be done with it. They have been very well described and organized on our webpage. So I think that's coming in. I can just jump in there. There we go. So I realize right now that I forgot to update the screenshot. I'm going to do this before publishing my slides, of course. But if you go to nfdiforbiodiversity.org, in this menu that here looks very short, actually in the live version, it's much longer. And one of the points is services. And inside these services, you will see everything that we offer completely publicly without any reservations. Behind this is a service portfolio. So there is a bigger list of more than 80 or 90 services that all our partners have in place, are offering in general. And the reason why you see only a subset is very simple. We want to describe the services in the same way, and we want to offer the same type of service, let's say, not only quality, but make sure that there's somebody available in the help desk to answer questions to this service, and that we make the same, we can give you the same information about what the service is, how it works, who is it for, and so on and so forth. And we also want to select, because some of the services might be very specific to a very tiny community, and we don't want to confuse everybody else with putting this on first. So we also need time to structure the whole thing. Okay, so the catalog is online. Visit that, check that out, but there's more in the background. So if you don't find what you're looking for, again, your best chance is write us a quick email, and then we can make a recommendation. I'm sure Daniel mentioned this already this morning. The NFDI in general is a huge network of 26 topic-specific consortia, and we work very closely with at least 3 to 6 of them because they're thematically very close. So microbiota, data plants, fair agro. So we know where to call and ask for additional help if we cannot give you the answers that you need. So I'm going to mention that we have collaborating workbenches, so systems that help you organize your data locally. So this would be, for example, generally installed at your working group and help you manage your data from day one. Installing, getting something like this in place is a hurdle. So we can help with this, but we also have some possibilities to give you an instance to play around with and figure out if this is something that you really need and want. The best thing here to do is to contact us, of course. Then we have a general data submission and brokerage service. So this is what I've been doing the last 10 years. It's basically saying we work together with 10 different long-term data repositories, 9 of them within Germany, 1 in the UK, that's the European Nucleotide Archive for molecular data. And we've built basically a single point of entry and contact for all of them. So basically you can drop your data and say, I know my data has to go to Pangea and ENA, can you make this happen? Or you say, I have a very complex data set, I don't know where to archive it, but you're the experts, what do you recommend? And we are going to recommend something. So we have templates in place. You get your DOIs. So basically part of the service is to say the right data to the right repository. So sequences to ENA, environmental data to Pangea, observational data to one of the collection centers. And if we split your data in different chunks, all of them will get a DOI or another persistent identifier, and we'll link those. So there will be like a network of your research output. And then it doesn't matter if people get in contact with the paper first or with the sequences first or with the environmental data first, they can follow the crumbs to all the other parts. And machines should be able to do this as well if we do our homework correctly. And all of these data centers work with embargoes. So basically you can say, I archive already today, and I actually make it available to the public in 3 months or in 6 months because I need time to finish my manuscript and I'm a little bit concerned about scooping, which I still haven't found a real example of in the last 10 years, but if you're concerned, this is always a possibility. We don't only get all the data in, we also have a unified data search that basically gathers the metadata from all these 10 data centers and presents it in a unified way. So technology-wise, that's not the big thing, but the big thing is getting all these different data providers with all the different data types to agree on a common set of features to present here and how to provide them to such a central resource. And you can also select this data and go and start visualizing it on a geographic information-based system. So anything that is geo-referenced, that has coordinates, you can start playing around right away with our visualization and analysis tool. So that's all I brought for today, and I really hope it was not too fast. Again, you're going to get the slides. Thank you very much for the attention, and keep in mind that was just a few highlights, many more services available. Go visit us, tell us what you think, tell us what you miss,